Thank you, Valeria. Thank you, Francesca, for giving me the opportunity to maybe present. Yes. All right. Yeah, to present the work that I've been doing at the Center for Neural Dynamics and MEG at the University of Tübingen on neural interactions across cortical layers. So this is the session on neural data. So I would like to start by describing the type of data that I've been working with in this project, which is laminar electrophysiology data, in which we record the activity of neural populations across all of the cortical layer, like all of the layers within the cortical column, so that in the end, our data has both a temporal as well as a spatial dimension over which it evolves. And we do this because we are interested in how different layers in the cortex talk to each other and exchange information. And we would like to measure this by computing some measure of the statistical relationships between the neural signals. And that's because, while on the anatomical level, we have a fairly good understanding of what the structure of the circuit is, at the functional level, especially when animals are behaving, the picture is not so clear. And so the major question that we try to address is whether, by taking measures of the functional connectivity on this spatiotemporal signal, is it possible to retrieve some anatomical features of the underlying neuronal circuit. Now, there are many measures for functional connectivity, but by far one of the more popular ones in neuroscience is Granger Causality, which is just model comparison method based on linear regression that asks how much better can I predict a given time series if I include in my regressors the past of another time series. And we can ask this in both directions, so we can also quantify the asymmetry in the interaction, meaning which signal drives which more strongly. It's a very popular method, but we know it has some statistical limitations, and what we argue is that in the kinds of recordings that we are dealing with, these limitations are made worse, they're aggravated. And so another question we try to address is whether Granger causality really is a valid measure for estimating connectivity between cortical layers. So to address these points, I went and analyzed part of a much larger data set collected in our lab of multi-area laminar recordings in macaque monkeys. I mostly focused on the prefrontal areas from which I extracted the local field potentials at every channel along the cortical column. And with these, I compute the Granger, like the pairwise Granger causality between each pair of these channels, so that in the end, I get these kinds of interaction matrices where you have basically channels on both sides, and this gives you an idea of how every channel in the probe is predictive of every other channel in the same probe. So we are within area. There is no across-area interactions in this work. And one feature that I'd like to point out is that the upper triangular component of this matrix reflects downward projecting interactions, so from superficial to deep layers, whereas the lower triangular component goes in the other direction, so from deep to superficial layers. And I point this out because I think it's evident by just looking at this example matrix that the lower triangular part is has a much stronger Granger causality signal than in the other direction, implying that in this data there is a very strong upward biased asymmetry along the cortical column, which is something that we find across all of the recordings that we have, across all of the prefrontal areas that we've looked at. And looking in the literature, there's a few studies that have done similar kinds of analysis, and they also report this asymmetry, yet nobody really tries to explain where is it coming from. So that's kind of where we try to come in. And we try to explain this asymmetry using a source mixing model in which we imagine that there are one or more independent neural populations without any interactions that project their activity onto a set of measuring channels with some noise. By analyzing this model and running simulations on it, we can show that an asymmetry like the one in our data can actually is guaranteed to exist even if there are no actual underlying interactions in the generative model, which means that at least partially our data could reflect this kind of noise-driven asymmetry rather than through interaction. So to get a better understanding of how our data is affected by this, we apply the matrix factorization method to these laminar Granger causality matrices that decomposes them into a sum of non-negative components. When we apply this to simulated data where we control the position and the mixing of the populations, we see that the components retrieved align very well with the mixing coefficient that we impose by design. And when we then go back to the data and apply it there, we also see spatially well-separated components where the peaks of these components align well with the 3 major laminar populations estimated with other neurophysiological methods. So in conclusion, we can say that yes, Granger causality can give us information about some structure of the circuit, but if we really want to use it to tease apart the interactions, we need to account for the source mixing effects that might obscure through interaction. So with that, I would like to thank Marcus and Konsti for supervising the project and the rest of the team, as well as you, the audience, for the attention. Thank you.